Question number 15. How many reductional divisions are required to form 400 synergids? Options: A 400, B 300, C 200 and D 100. This is a question related to the reproduction of the plants. So here this is starting with the megaspore mother cell. In the female structures, that is in the ovule, inside the ovule we can see the presence of a diploid tissue called as a nucellus. If you consider this as the ovule, this structure, these are the integuments, and this structure is called as a nucellus. Here inside the nucellus, one cell will be converted into megaspore mother cell. That megaspore mother cell, consider this as a megaspore, this is a nucellus and megaspore mother cell. It will divide, it is diploid cell actually. This will divide by meiosis and forms into a linear tetrad of megaspores like this. And here 4 haploid cells will be formed. And out of this, the upper 3 will get degenerated, only the functional megaspore will be left out. So on the lower side, this one, this is called as a functional megaspore. So this functional megaspore will be haploid because this megaspore mother cell, which is diploid, had undergone meiosis to form tetrad of megaspores. These tetrad of megaspores are also haploid in condition, and the functional megaspore is also haploid in condition. This functional megaspore will undergo further mitotic divisions, and this will be converted into embryo sac like this. So on the micropylar region, they have egg apparatus. This is egg cell. One egg cell will be formed and 2 synergids will be formed. Whereas on the antipodal side, there we can see the formation of 3 similar type of cells called as, shall as I said, 3 antipodals will be formed, and in the center 2 polar nuclei will be present. So here, that means when one megaspore mother cell is undergoing one meiosis, it is leading to the formation of 2 synergids here. That is, one reduction division is leading to the formation of 2 synergids. Formation of 2 synergids. And in the question it is given as to produce 400 synergids. The number of meiotic divisions which are required are, that is 400 into 1 by 2. It comes to the meiotic divisions which are required to produce 400 synergids or 200 meiotic divisions. So from the given option, option C is the correct answer. Question number 16. The ratio of chromosomes moving to each pole during anaphase 2 to those moving to each pole during anaphase 1. The given options are A 1:1, B 1:2, C 2:1 and D 1:4. So chromosomes moving to each pole. So during anaphase 2 and anaphase 1, here whatever the chromosomes which are moving to opposite each pole, consider this as anaphase 2 and this is for anaphase 1. Here in anaphase 1, what happens is the diploid condition is converted into haploid condition, So for example, if 4 chromosomes are participating here, the same number of chromosomes will be there in the anaphase 2 also. That means if 4 chromosomes are moving to each pole, here also the 4 chromosomes are moving to each pole. That means the proportionate ratio will be 1:1. So from the given options, option A is the correct answer. Question number 17. What type of cell division takes place in the functional megaspore initially in angiosperms? Options: A. Homeotype without cytokinesis. B. Reductional without cytokinesis. C. Somatic followed by cytokinesis. And D. Meiotic followed by cytokinesis. So here the megaspore mother cell will undergo meiosis first and converted into linear tetrad of megaspores. And these, out of these, the upper 3 will get degenerated and lower functional megaspore will be present. So which is haploid in condition. This functional megaspore, what happens is the nucleus present in this one, first it will divide. Consider this as a functional megaspore. The nucleus will divide and these two nuclei will move towards two opposite poles. Only the nucleus will be dividing, and the nucleus which is present in the functional megaspore will be haploid in condition. So definitely it have to undergo mitotic division, and this nucleus further divide to form into two nuclei on one pole and two more nuclei on the opposite pole. So here only the nucleus will be dividing and leading to further change where the two nuclei present on upper pole will divide to form 4 nuclei by mitosis, and the two lower one will divide to form 4 nuclei. So in this one, if you could see here, in all the stages the number of cells will be same, that is the number of cells are retained as 1, but here the nucleus number is increasing. So here the cytokinesis is not taking place, that is division of the cytoplasm is not taking place, only the nucleus is dividing. The nucleus is dividing by a process called as mitotic division. Mitotic division is otherwise called as equational division or the homeotypic division. Why? Because the chromosome number will be same from the parent to the daughter, and between the daughter cells also the chromosome number will be same, and whatever the nature that is present in the parental cell, the same type of characters can be seen in the daughter cell also. That's why we can call this mitosis as homeotypic division. So in the functional megaspore, homeotypic division will occur, but without cytokinesis. So from the given options, option A is the correct answer. Question number 18. Colchicine interferes with options: A. DNA replication, B. Organization and orientation of spindle, C. Chromosome condensation, and D. None of the above. So here colchicine is a chemical which is derived from Colchicum autumnale plant. This Colchicum autumnale belongs to Liliaceae family, and colchicine have special functions to be considered, that it is acting as a chemical mutagen. This is one function, and at the same time it is a one which will obstruct the organization of the spindle. That is one, that is organization of the spindle fiber will be obstructed by this one. and when the spindle fiber organization is obstructed, the chromosomes will not be oriented to the equatorial region. So not only spindle organization, they will obstruct the organization of spindle fiber, but also they will obstruct the orientation of the spindles. But it is not concerned with DNA replication nor with chromosome condensation also. So from the given options, option B is the correct answer. Question number 19. Daughter cells formed as a result of meiosis are not similar to that of parent cell because options A. Meiosis is completed in 2 stages. B. Prophase is the longest phase. C. Nucleus size increases in daughter cells. D. Crossing over takes place and chromosome number is halved. So here, during meiosis, particularly during meiosis 1, what happens is the first stage called as prophase 1. In this prophase 1, as it is the longest phase and the many such changes are taking place during this stage. Apart from this one, here if we talk about actually here 5 stages are there. Those are called as packetine, zygotine, and like different stages are there. So out of this, if you take a packetine stage, in this packetine stage, what happens is the changes of chromosomes will be taking place. So actually before this one, there is one more stage is called as zygotine. And during these stages, what happens? The crossing over is taking place. Crossing over means the homologous chromosomes which have been attached with each other, those homologous chromosomes like this. This is the one homologous chromosome. And the other homologous chromosome. So here we can see the pairing of chromosomes will be taking place, that is called as synapsis, and at this point where synapsis is taking place, these fragments will get broken and exchange of the fragment pieces will be taking place. If for differentiation we can change the colors here. Let us take one as a black and one as a white one. So like this, and this broken part will get exchanged to this one, this broken part to this one. So it appears to be like this. Here we can see that this part have been taken from this non-homologous chromosome and this part is taken from the non-homologous chromosome. Likewise, the chromatid pieces are exchanged between them, that is called as a crossing over. So this crossing over will occur during the packetine stage. During anaphase 1, the chromosome number will become half, that is the genomes which are there, that is 2 genomes which are present here, they will get these two are the genomes, this is N and this is N. So combined we call it as a 2N condition. The 2 genomes will get separated. So because of that reason, so the daughter chromosomes are different from that of the parent cell. So from the given options, option D is the correct answer. Question number 20. Choose the correct match. List 1. A. V-shaped chromosome. B. L-shaped chromosome. C. J-shaped chromosome. And D. I-shaped chromosome. List 2. 1. Terocentric. 2. Metacentric. 3. Submetacentric. And 4. Acrocentric. The given options are A. 4, 1, 3, 2. B. 3, 2, 1 and 4. C. 1, 4, 3, 2 and D. 2, 3, 4 and 1. So let us take the list on one side, list 1. Here the shapes of the chromosomes have been given. Generally the chromosomes will acquire different shapes during anaphase, where V shape, L shape, J shape and I shape. Here when they acquire different shapes, so here in the V shape structure, the centromere is exactly present in the center and the two arms are equal in size. This can be seen in the metacentric. That is, metacentric have been given as 2, and L shape can be seen. So here we can see the shape like this. Two equal halves will be present, and in L shape the shape will be like this, where one arm will be very long and one arm will be short. So here this is a condition where submetacentric chromosome will exhibit this character. That is the third option here, where the centromere is near to the center and one arm will be long and one arm will be short. Whereas J shape, where the one arm is very long and one arm is very short. So this can be seen in the acrocentric chromosomes. Acrocentric chromosome, that is option number 4, and that is the centromere is near to the tip. And the last one is I shape, where the centromere is exactly at the telomere region, that is in the telomere, so we call it as telocentric chromosome. So likewise the chromosomes will acquire different shapes during anaphase, so depending upon the position of the centromere. So from the given options we can say that V shape is exhibited by metacentric, L shape by submetacentric, J shape by acrocentric and I shape by the telocentric chromosomes. So 2, 3, 4, 1 are the correct sequence of options. So from the given options, option D is the correct answer. Question number 21. Choose the correct match. List 1. A. Zygotine, B. Pachytine, C. Diplonema and D. Diakinesis. List 2. 1. Crossing over, 2. Terminalization, 3. Formation of bivalent and 4. Repulsion of paired chromosomes. The given options are option A 1, 3, 2 and 4, B 3, 1, 4 and 2, C 2, 4, 3 and 1, option D 4, 2, 1 and 3. So here let us take the options that have been given here. First one, zygotine. Zygotine is a process where the bivalent formation can be seen. That is, bivalent means dyad will be there. That is, the synapses will occur during this stage. Synapse is a process where the two homologous chromosomes will pair with each other like this. So now this is a maternal chromosome, this is a paternal chromosome. That is, the homologous chromosomes will be formed because of the synapses or the pairing. So here synaptonemal complex is the one which is helping in the process of synapses. So now this structure, whatever that have been formed is called as a bivalent. So here the bivalent formation, the option number 3, formation of bivalent. And B, that is pachytine. Pachytine is a process where after the formation of bivalents, what happens is these bivalents will split longitudinally and forms into tetrad like structure like this. We call it as pachytine tetrad, and these two chromatids are called as sister chromatids and these two are called as sister chromatids. and one of the chromatid from one homologous chromosome and one of the chromatid from other homologous chromosome are called as a non-sister chromatids. During packeting, the non-sister chromatids will get attracted with each other and they will form a pair, or we can say they will form an attachment like structure. So we can show like this. And this is called as chiasmata, and this part will get broken, and whatever the part that have been separated will be transmitted to the other non-sister chromatid. Like this exchange of the chromatid pieces will be taking place. The exchange of chromatid pieces is called as crossing over. That is option number 1. And C, diplonema. The next stage after packeting is diplonema, where till now up to this packeting stage there is an attraction between the chromosomes, but here the synaptonemal complex will gradually dissolve and the attractive forces will also gradually decrease, and at the same time the repulsion between the chromosomes will occur, and because of that one, what happens, they will try to move towards the opposite side. So repulsion of paired chromosomes will occur. That is option number 4. And D, diakinesis. During diakinesis, what happens is the repulsion will further increases. As the repulsion further increases, what happens? The chiasmata that is present here will gradually move towards the tip like this. Now the attachment will be only at the tip region. So this is called as a terminalization. So these are changes which are taking place during the 4 different stages of prophase 1, that is zygote, formation of bivalent, packeting, crossing over, diplonema, repulsion of paired chromosomes, diakinesis, terminalization process. So from the given options, option B is the correct answer. Question number 22. Identify the correct ascending sequence of the following aspects of meiosis. A, number of spindle apparatae formed. B, number of chromosomal divisions. C, number of cells formed. And D, number of nuclear generations. The given options are A, B, C, A and D. B, B, D, C and A. Option C, D, C, A and B. Option D, B, D, A and C. So let us start with the first one, that is number of spindle apparatae formed. During it is the question related to the meiosis. In meiosis, as we know that 2 times the nucleus will divide, and during the process, that is during meiosis 1, 1 spindle is formed, whereas during meiosis 2, 2 spindles have been formed. So total number of spindles that have been formed will be 3. Spindle apparatus will be 3 here. Then next one, number of chromosomal divisions. So the chromosomes will divide only once. Then number of cells formed. So as a result of one meiosis, the number of cells that have been formed as a result will be 4. And D, nuclear generations. So here there are 2 nuclear generations, one is haploid generation and the diploid generation. So that will be 2. So from the given options, ascending order. So B will be 1, D is 2, A is 3 and C is 4. The correct sequence is B, D, A and C. So from the given options, option D is the correct answer. Question number 23. Arrange the various phenomena of meiosis in a correct sequence. A. Pairing of homologous chromosomes. B. Separation of genomes. C. Recombination between two chromosomes. And D. Chiasmata. The given options are option A, D, B, A, C, option B, D, C, A and B, option C, A, C, D and B, option D, C, D, B and A. So here in this process they had given A, B, C, D. So from the given options we can say that first thing, that is pairing of homologous chromosomes. The pairing of homologous chromosomes is nothing but the synapses. So this process will occur during zygote, and zygote is the part of prophase 1. Then recombination between the two chromosomes will occur. That is, recombination means that is a crossing over will occur where the broken fragments of the chromatids will be exchanged between the non-homologous chromosomes. That is a recombination between the two chromosomes. That is during packaging. After zygote, packaging will come. So that is also the part of prophase 1. Then next one will be the chiasmata. That is after the recombinations. What happens? The chiasmata will be clearly being seen during the next stage. That is what happens is during diakinesis stage, the pairing of chromosomes or the attraction between the chromosomes will gradually decreases and repulsion will increases. As the repulsion will increases, the chiasmata can be clearly be seen in the form of a X-shaped structure. So that is during the diakinesis stage. So before this, this can be clearly been seen in the diplonema stage, and of course it will be moving to the tip. So chiasmata will be clearly seen during the diplonema stage, and the next one will be the separation of the genomes. That is whatever the diploid condition is present in the non-homologous chromosomes which have been there, so they will get separated. That is what we call it as a separation of the genomes, which will occur during anaphase 1. So these first three are taking place during prophase 1. In the first stage, zygote, packaging, and the next one diplonema. After diplonema, diakinesis will occur, and thereafter the metaphase 1, then anaphase 1. So this is a sequence of changes which are taking place during the meiotic phase. So the correct sequence is A, C, D and B. So from the given options, option C is the correct answer. Question number 24. Choose the correct match. List 1. A. Anaphase 1. B. Anaphase 2. C. Metaphase 2. And D. Telophase 2. List 2. 1. One spindle apparatus. 2. Separation of two genomes. 3. Two spindle apparati. 4. Separation of two chromatids. And 5. 4 daughter nuclei. The given options are option A, 5, 4, 3 and 2. Option B, 2, 4, 3 and 5. Option C, 1, 4, 5 and 3. Option D, 4, 2, 1 and 3. So here let us start with the first one, that is anaphase 1. Anaphase 1 is the most important stage where the genomes are separated, that is whatever the homologous chromosomes that have been two homologous chromosomes which have been paired, so they will get separated during this stage, that is the separation of the genomes will be taking place. The haploid number will be formed, that is the diploid nuclei will be divided to form haploid number. This can be seen in option number 2. Second one, that is anaphase 2. During anaphase 2, the centromere will split. The centromere will split, and how does the centromere will be splitted means because of the formation of the two spindle apparatus. The two spindle apparatus for two separate group of nuclei which are present in the same cell. The two spindle apparatus will come and attach to the centromeres of the chromosomes. Then they will contract to divide the centromere into two parts. When the centromere is divided, two chromatids which have been separated out will be moving towards the two opposite poles. So here two spindle apparatus will be formed, and separation of the with the help of two spindle apparatus, separation of the chromatids are will be taking place. So from the given options, option 4 is a correct one. Then C, that is metaphase. Actually during metaphase 1 only one spindle apparatus will be formed. So whereas after the completion of meiosis 1, the two nuclei will be formed. The two nuclei will be separated, and that is during prophase 2 the chromosomes are released into the cytoplasmic content, and each of them will get separated because of the spindle apparatus. So here formation of two spindle apparatus. Is the one, that is option number 3. And D, that is telophase 2. Here telophase 2 is the last stage of meiosis 2, where two daughter nuclei are divided to form into 4 daughter nuclei. That is option number 5. So the correct sequence of options are 2, 4, 3 and 5. So from the given options, option B is the correct answer. Question number 25. Identify the labeled parts of following diagram. The given options are option A, A1, A2 are sister chromatids, B1, B2 are non-sister chromatids, D is centromere, C is chiasmata. Option B, A1, A2 are sister chromatids of non-homologous chromosomes, B1, B2 are sister chromatids, C region of crossing over, and D centromere. Option C, A1, A2 are sister chromatids, B1, B2 are sister chromatids, C is crossing over, D is centromere. Option D, A1, A2 are sister chromatids, B1, B2 are sister chromatids, D is kinetochore, and C is centromere. So here this is a diagram related to the question here. So these two are the chromatids, that is A1 and A2, and these are the part of one homologous chromosome. That means and at the same time B1 and B2, these two are also the chromatids of the same homologous chromosome. That means here A1 and A2 are the sister chromatids of non-homologous chromosomes. Here B1 and B2 are also the sister chromatids. And C is a region where the crossing over is taking place. That is the part of chromosome chromatid A2 is exchanged between the B1. So this is a part where the crossing over is taking place. And D is the centromere part. So from the given option, we can say that option B is the correct answer.